This is This Week in Science and Education, episode 27, Science is Visual. It's time for This Week in Science and Education, the show that talks about all things science-related and with an educational focus. How do we take that information, help teachers bring it into the classroom? On today's show, Science is Visual, pictures about science. All that and more coming up next on This Week in Science and Education. This Week in Science and Education is brought to you by Polycom, world leaders in unified communication. Check out polycom.ca. And by Muley Systems. Need a backup? Have some important files and documents you don't want to lose? Check out muleydata.com/vrock. Hey everybody, it's time for This Week in Science and Education. This is your host, Kevin Cougler. On today's show, a couple of exciting things. But first, before I get to that, joining me as always from the Kawartha Pine Ridge District School Board, Mr. Colin Jagoe.
Hello. How's it going, Kev?
Good, Colin. What's new up there at the school board today?
Well, it's first day back after a long weekend, and it's the typical first day back after a long weekend, running around like mad, doing all sorts of things. But it's good to be here.
Yeah, I hear what you're saying. It definitely feels like we've had too many days off. It's been a comedy of errors today, trying to get shows done and just banging our heads against the wall technically. So, and again, we do apologize for the show being late today. It's 2:27 instead of 2 o'clock. But for those of you that have stayed with us, we thank you for doing so. And hopefully everything will be smooth sailing from here on in. And speaking of smooth sailing, it's that nautical engineer, that mad scientist himself, Dr. Thomas Merritt from Laurentian University.
Hey, Kevin, how are you?
Well, I give you that big lead up and that's all you got. Hey, how are you?
Yeah, what are you going to do? It's the best I can do given the circumstances. It's great to be here. It's nice to see the two of you again this afternoon.
Lakes are still frozen up there. There's no sailing to be done just yet.
Yeah, not a lot.
Actually, I was kind of tongue-in-cheek about that, but you are a guy that knows an awful lot about the water because of your, everything you do with the rowing and whatnot. Actually, do you want to just talk about that for a little bit? Because you've had quite an interesting experience with the rowing club up there. Just tell everybody what you're doing with that.
Well, I run a rowing program for people with disabilities. Rowing, we call adaptive rowing, so adapting the sport of rowing to people with various disabilities. We started the program here in 2007. It's going extremely well. We've had a few dozen athletes through the program over the last 4 years. We work primarily with athletes with physical disabilities, or at least that's how we started the program, and we've moved into a lot of intellectual and developmental disabilities as well. In 2008, one of our athletes went to Beijing with the Paralympics. That was really quite a feat. Steve is a retired military sergeant, was disabled, was injured in a parachuting accident while he was training with the military. And I think 3 years out of his injury made it to Beijing as Canada's arms and shoulders rowing, which was an amazing feat for him, that kind of rehab from paralyzing accident to this international stage of one of the most accomplished athletes in the world. That was quite an achievement. It was really neat to be part of that. The community program here is extremely active, and right now we're working with local high schools with classes with special needs. So I've got a couple of classes for children with autism spectrum disorder or some different learning disabilities, and they come out and row with me. So we do that through the year, and as Colin mentioned, the water's frozen a lot up here. So we spend a lot of time on rowing machines, but it's a really neat way to get people active, and the rowing machines are really easy. There's a lot of technical issues with getting somebody who's paralyzed in and out of a boat and on the water, and the issues are a lot easier. You're just working with rowing machines. And when we're coaching athletes with learning disabilities or developmental disabilities, simplifying things is a big deal, and so starting on the rowing machine is a great way to get that started. We've had fantastic support from Rowing Canada, the national body that supports rowing, and Row Ontario, the provincial body that does that. And we've had grant support from Parasport Ontario, another group that's been really supportive of adaptive rowing in general and of this program in particular. So yeah, that's been a really big part of my last 4 years, getting that program together and keeping it up and running. We just received another grant from Rowing Canada 2 weeks ago, 3 weeks ago, within the last month. And so we're always trying to expand the program and try something new. This year we're going to try to get some of the athletes out in singles, give them a little bit more independence. And so we're trying to buy a boat that's essentially indestructible. You know, you get kids out in equipment, and if they don't know what they're doing, and if they have issues with being coached, it's a little bit nerve-wracking if you've got them in a $14,000 boat and they can crack it in half at any second. So we're going to buy a plastic boat that in theory they can't break, and turn them loose and say, okay, have a good time with us, see how it goes.
That is a really worthwhile thing that you do. I really applaud you for doing that. You know, you reminded me of a book that I just got done reading. I'm trying to pull up the title here, which is why I keep going over to my iPad. But the book is written from the perspective of somebody with autism. And it's something about the upside down dog or something like that. And I'll get it for you in a minute.
You're not talking about the murder mystery where it's actually written from the perspective of the dad and his son has autism. The dog in the night.
Dog in the night or something. The dog.
Yeah, but it is written from the perspective of the son,
Okay.
and
Yeah.
there's a murder, and the son with autism tries to solve it, and it's. I just got it done on the weekend. I couldn't put it down. I read the whole thing in 2 days. It was absolutely fascinating, just to have that that introspection into how their mind works, and and how their lives are mathematical, and everything has to be in a certain place, and how do you overcome some of those challenges? How do you work with those challenges with rowing? Is it how does that all work?
That's a good question. It works differently with each athlete, and I say that every time I talk about adaptive rowing, I bring home the fact that every athlete is unique, and that's true whether you're talking about autism, whether you're talking about learning disabilities, whether you're talking about physical disabilities. And the truth of the matter is that's true when you work with able-bodied athletes as well. But the differences are differences that we're all more familiar with and not unexpected when you come in. So as a coach working with athletes that have different needs or different requirements, it's really just a matter of taking a step back and saying, what does each athlete need? And that's exactly what we do with the able-bodied athletes. There are differences when you work, and maybe extremes, when you work with people that are paralyzed, paraplegic, quadriplegic, when you work with different learning or developmental issues. A lot of the stuff with coaching the students that have a learning disability or they have a developmental disability, we scale things down a little bit. You change your expectations and you gear the expectations to the athletes. One of the things that we did last year was we had a couple of athletes that had panic disorders as well as other learning or developmental disorders. And with them, one of the athletes, the big accomplishment for the summer was getting her to walk out on one of the docks. And the docks are not fixed. They rock a bit on the water. And she didn't want to go. She wasn't comfortable on the dock. She was comfortable working with me. She was comfortable working with her teachers. And we got her to the point where she was comfortable walking out of the dock. And honestly, the biggest help with that were the students that are working with her in the class, her colleagues in that class. And they were all excited to come out and stand with her on the dock. So, as a rowing coach, maybe 5 years ago, if I thought about what I was going to be doing, I wouldn't necessarily have told you that a big accomplishment for the program would getting somebody to walk out and stand independently on a dock. But, that was a really big accomplishment. So you scale the expectations to the athletes. I work with the athletes an hour, 2 hours, once a week. When we were trying to get to Beijing, we were a couple hours a day, 5 to 7 days a week. But it's still, the people that really do a great job are the teachers that are working with these kids 9 to 3, 5 days a week in all these different programs trying to get the kids engaged. So I do one little bit once a week with these kids. The teachers are out there day in and day out. They come to see me, they go to the Y, they have other programs that they're involved with. So they do a fantastic job. I've got a short period of time with one little skill set that I'm going to try to get them more active, and we do the best we can with that. It's fantastic. It's a lot of fun. It's hugely rewarding. But I can't say enough about the teachers that make that kind of program work.
We applaud you for those efforts. It must be very rewarding actually to do that. Anyways, let's move on with the show, gentlemen. Our first item, I was kind of surfing the web on this new app that I got. It's just new to me. It's not new out there, but it's the Pulse app, which is basically an application on my iPad that allows me to pull in different RSS or news streams from different sources. And the one that came in through Wired.com talked about different science visualizations. And I thought this was interesting from the perspective that, you know, we often talk on the show or we, you know, in the classroom, Colin, you know, about different things that are, you know, science related, of course, but this actually, for me, it showed the beauty of science and the interesting graphics that you can come across when you're working in the scientific realm. So it's Wired.com is the name of, or is the address of the website, and then the search string for those of you that are interested is stunning science visualizations, the year 2010. And I think Colin's going to be able to pull up some of these pictures for you.
Give me a second here.
So while we're doing that, I've just
Yeah,
a couple of comments. There we go. Yeah.
Yeah.
Great. So, what are we looking at with that one?
You know what? Before we start, I think Thomas was going to make some comments, but I've got comments as well, because one of the things I like about some sciences in particular is the visual nature of them. Like, I love astronomy as a science, as an interesting science to think about, but the visual nature of it is amazing, right? To be able to pull information out of photos that are actually just stunning to look at has always appealed to me. So I love science visualizations from astronomy point of view, but we're going to look at some that are, you know, that are on much smaller scale, down to, you know, virus size and molecule size, that are equally as amazing. So I don't know if, Thomas, that was sort of some of the stuff you were going to talk about or not, but that's what I think about some of these visualizations.
What I was going to talk about, just sort of taking this to the perspective of a working scientist. The article that accompanied these great images was talking about the idea that too often science is lost in black and white and a series of numbers and tables and figures, and that given the hand of a talented artist, you can really bring a lot of things to life, and that is absolutely true, and I think it's an enormous part of bringing science into a classroom. But one of the things that I've learned as a working scientist is to graph and visualize all of my data. And so anytime I'm doing an experiment, and now as a professor, I don't get to do as much time in the lab, and so it's working with my students. And I just basically tell all of my students, anytime you do an experiment, graph out the results. Yeah. And it's not as much in that case trying to be pretty, it's just trying to visualize it. And as a species, we're really good at processing visual data and looking for patterns and finding patterns in visual data. And if the data is just a list of numbers, it's very difficult to see those patterns. So this kind of visualization can bring science to life and get people excited because it's so pretty. But as a working scientist, I assure you, this allows us to see patterns in our science that we miss otherwise. And the standard story that I tell about my own lab, I was doing a set of assays where we were looking at the effect of temperature on enzyme function, not something you would really think of as being something pretty to visualize. And I had published these results. I had presented these results in a number of different conferences and at different talks. And I was finally reworking my talk, and instead of just having a table of numbers, I graphed out the figures. And I realized there was a pattern there I'd missed the whole time. And we went back, redid the experiment, increased the sample size, increased the replication, and lo and behold, there's actually a really interesting story there. And we'd missed it. I had missed it because I was looking at a column of numbers. And we had predicted that, given the structure that we thought we had for the protein, that the pattern should be this. There should be a low stability, a high stability. And the numbers said there wasn't. And we graphed it out, and we realized that the pattern was exactly what we predicted. It was just there was a lot of variation. And when we cut down on the variation with better experimental technique, lo and behold, it's statistically significant, and we're trying to publish that paper now. So this visualization is a great way to get people engaged in science. But one of the things you can tell your students is that we use this as scientists all the time. This is how we as a species process data, by visualizing it, by looking at it, by looking for these visual patterns. And so there's a lot to be said for these kind of visualizations, not just as learning tools, but actually as scientific tools as well.
We should probably point out that these images are part of the 2010 International Science and Engineering Visualization Challenge, which appeared in the February 18th issue of Science. Sorry, Colin, you wanted to jump in.
No, No, that's fine. And I was going to talk about this one a little bit too, but because this is an example of one of those things. But then Thomas made me think about something else. This is a model of an HIV virus. This is the human immunodeficiency virus, sort of modeled in 3D, so you can sort of get a really good look at, or a really good feel for what it looks like. But you talked about the visualizing in humans being good at doing things that, looking at numbers you can't necessarily pull out. It reminded me of talking to the guy from Foldit a few months back, and how people are good at finding those patterns much quicker than sometimes even computers are. So forget looking at the numbers, just by playing with the protein folding game in this particular instance. So it's really interesting. So these visualizations have a lot of interesting applications. And when you look at this one here, not being someone who really knows much about the HIV virus, but this is interesting, right? This is a 3D model, but you can start to look at all of the different bits and pieces in there. And I don't know if you guys can see my mouse or not, but like even talking to kids here, you can get an idea of the layered structures and the various bits and pieces that are in there. And it makes me as a learner go, well, what is that? What is that supposed to represent? Which then gets you into the actual science of it. So this is a starting point to get interested in digging deeper into something. So I think it's really, really cool.
There's another one, just a few over there, Colin. I don't know if you can switch to that one. It's the nanoscale monolayer.
Sure.
It's a couple over. It's kind of blue with
that one.
Little lights or whatever.
Yeah, this one.
Yeah, exactly. That is absolutely gorgeous. Maybe talk a little bit about what that image is.
I don't remember. You seem to remember.
I've got it here in front of me. I'll tell you. Actually, I'll just read a little the caption that's underneath. That probably does it the best justice. So this is a nanoscale monolayer, and the caption reads, "Lending artistic vision of the nanoscale world, two researchers bore down on a gold surface with an atomic force microscope." That's impressive in itself. Atomic force, I love that word. Sitting on top of the gold was a nano-thin layer of self-assembled molecules. The rippling blue that you see throughout that image result earned the first place posterity in the photography category. So congratulations to them. And that was Seth B. Darling from Argonne National Laboratory and Stephen J. Sibner from University of Chicago. So that is quite a striking image. This is sitting on top of the gold. This is a layer sitting on top of gold. Doesn't look gold at all, does it?
No, but I'm assuming that's the photography of it. I'm guessing what this is they use the gold as a substrate to put, materials down that then become these self-assembling molecules for creating new materials. It could be anything. It could be drugs. It could be, I don't know what it is, but it just shows how we use certain materials and certain techniques to then make things. Like some of the people we've talked to in the past talk about assembling molecules and that sort of thing. And this is using one form of this molecule assembly at a nanoscale to try and make some things that we've not made before, right? And it's a neat picture, but it shows the surface of the gold and the sort of the 3D leveling of the various parts of the gold, and that they're being used then as sort of the bowl, if you will, which they use to cook up new materials, which is really, really neat.
Hey, do you want to see what a tomato seed looks like up really close?
Which one is that?
Second one from the end there.
Oh, there it is, with the hairs on it. Yeah, that's a cool one too.
Tomato seed hairs. Very cool. This is trichomes, I guess is how you pronounce it.
Just trichomes.
Little tiny hairs. Sorry?
Just trichomes, yeah.
Trichomes? Okay, they secrete the goo that encases them, so I guess that's what we're looking at there. What a fantastic image.
Now, this is really interesting, because in our classes, we've been part of a project from time to time called Tomatosphere. Are you familiar with Tomatosphere, either of you guys? Which is a project of the Canadian Space Agency, which engages basically grade 6 students and grade 9 students in growing tomato seeds. And what they do is they give the tomato seeds various treatments, usually simulating some sort of space environment. So whether it's on the surface of Mars, whether it's exposed to the vacuum of space, whatever. So they actually send tomato seeds up in space, either on the space shuttle to the International Space Station, that sort of thing, and then bring them back and dole them out to high schools all across Canada. And kids then grow these tomato seeds with controlled tomato seeds, and the results are all then pooled back in a central database, looking to see if these various conditions affect germination and growth rates of tomatoes. tomatoes, right? So, tomatoes from space. And it's a really neat experiment because it's hands-on. The kids are really interested in it. But when you look at this picture, right, it starts to give you some of the structural indications of how hardy a tomato seed is, right? It doesn't dry out because it's got basically a layer of fur around it, right? And it's got some of this protection from things like ultraviolet radiation in open space and in the vacuum of space. So it pulls together something that you may be doing in your class with a really neat picture that shows why tomato seeds are really, really hardy. So this is, again, a neat picture with a lot of really cool connections.
Coming up, we're going to talk about human DNA contamination that some scientists have seen in genome databases. Thomas is going to tell us all about that. But before we get to that, I do want to mention our friends over at Mueller Systems in Woodstock, Ontario. They are the company that helps us protect all of our data. So those pictures of the grandkids or the children or the important files that you have on your computer you just don't want to lose. These are the folks that help us protect that data. So if you go to the website today, you'll be able to download a free trial, which is www.muellerdata.com/vrock. And we hope that you do visit the website. 90 days is a long time and a good amount of time to be able to test out a product. Download it, install it, configure it. If you run into any trouble, all you have to do is call them up. They'll actually answer the phone with a live human person that will help you figure it out. And if that wasn't enough, they'll actually make sure that it works for you. So try that on one of those, cloud-based backup services. You never get them on the phone. Who knows where they are? Anyways, muellerdata.com/vrock. We do thank them for their support. Dr. Merritt, human DNA contamination seen in genome databases. Very interesting story. Tell us all about it.
Yeah, it is really interesting, and it's one of those things that I think we've sort of seen on the horizon that this kind of thing was going to be coming, and it was just a matter of trying to wondering when it was going to hit. And Colin, were you able to dig up the details?
Yeah. Yeah, I've got it here. Just and I'm reading it because Thomas doesn't have it in front of him here, just so you know. But the story on the New York Times from last week, I guess February 16th, but talking about how these researchers have found that nearly 20% of the genomes held in computer databases are contaminated with human DNA. Right, so things like that they use for model organisms for crops and genetics research, that sort of thing. They mentioned some roundworms and some frogs and some other crop plants, but these researchers were digging into the genomes of these particular model organisms and found human DNA in amongst the DNA that was supposed to be representative of their species, which is probably not a good thing, but it's something that hadn't been really noticed before as to how much they were contaminated. And they talked about things like just skin cells in the preparation of these things for storage and that sort of stuff can contaminate these samples. So interesting, and I know, Thomas, you've got a lot more on sort of the ramifications of what that means from a genetics point of view.
Yeah, well, from a genetics point of view, but also from a biomedical point of view.
Sure.
So essentially what they found was if you go in and look at these international databases, and anytime we publish a genetic sequence, anytime somebody does a study that's funded by the government, you have an obligation to take the information, the DNA sequence that you have read, and deposit it in these international databases so that other scientists can work with it. And so I do this anytime I publish a study. The sequences, the DNA sequences that I've based that study on, they get published in something like GenBank, and it's an international repository. Anybody has access to it, and that's part of the game of science, is getting the information not just for your own work, but for everybody's work.
Right.
And one of the issues with these databases is they're only as good as the stuff that goes into them, and this has always been a case. I spent almost 6 months working with one gene that in fact was another gene, and it was misannotated in the genome, in the databases, and so we had been pursuing this thing as what we thought was an A gene, and in fact it was a B gene, and we found that out 6 months into the project, and luckily the next step was to look at the B gene, and so we were 6 months ahead on project B, but we hadn't gotten started on project A. What's happened in this particular case is there are the submissions in the database that are supposed to be things like C. elegans, which is this worm model species, and we use C. elegans to understand genetics in a very broad sense. It's a very powerful developmental and genetic tool. And when they actually looked at the individual sequences, a certain fraction, and Colin, you just gave us the fraction, wasn't C. elegans DNA.
Yeah.
So, and 20% is frightening. That's really high.
Sure is.
And it was actually human DNA. So how does this kind of thing happen? We used to do DNA sequencing and genome sequencing in a very what we call hierarchical fashion. You would isolate the DNA from an organism. You would clone that DNA into bacteria. We would literally cut the DNA into pieces. You would move the pieces into a bacteria, and they would be there in the bacteria. And then you would go through and systematically sequence what we call these clones, and then assemble it up into an entire genome assembly. In the past 5 years, a lot of that has moved from this sort of hierarchical sequencing to what we now call more of a shotgun approach, where you just take a lot of DNA and sequence it in relatively small pieces, and then we use computational tools to reassemble those pieces. And I don't know if the contamination that we're looking at is a function of having moved from this hierarchical procedure to the shotgun sequencing, but it's certainly a possibility. Either way, what's happened is you've got things that are not the species that you're working on that have contaminated into the sample. And so it's supposed to be fruit fly, or it's supposed to be roundworm, and in fact it's human. And you know, it's not just human, it's Bob the lab tech, and Bob got his fingers in it, right? And I've sequenced my own DNA at least once, and the lab I worked in for my PhD for a number of months, everybody in the lab was sequencing one of the postdocs' cytochrome B, because unbeknownst to us, he had contaminated everything.
Wow.
And so there was Doug everywhere. And so all the shark sequences were human sequences, and we never did actually manage to convince everybody to give a blood sample and do the genotyping, but we were relatively certain we know exactly whose cytochrome B everybody was sequencing. So, you know, this kind of thing happens. Now, what does it mean in this case? So with my own work, when it was the wrong gene, that just set us back 6 months. With sequencing Doug's cytochrome B, that just meant that we'd wasted those resources for a number of months sequencing the wrong gene. When we start working on these model species, it may be that it just sort of sets back that research, and you've got to go in and take a focused approach. One of the ways that we're going in modern genetics is medical genomics. And you know, we're going to be sequencing your genome, your genome, your genome, my genome, and we're going to be using that information to make medical decisions. Awesome. Fantastic. Wonderful thing to do. If you're working with roundworm DNA and suddenly something doesn't look right, it's very easy to distinguish human DNA and roundworm DNA. Right? So we go through and we know what we expect from C. elegans, we know what we expect from humans. And so you say, okay, well, we screwed up somewhere and we've got a contamination here. This is obviously a human gene sequence that's been labeled as C. elegans. We're going to throw that out. If you've contaminated one human DNA sample with another human DNA sample, how do you know which one is which, and how do you know there's contamination in the first place? If you're making a biomedical decision based on a gene sequence, and you don't know if it's the gene sequence of the patient you're working on or the technician who took those samples, that's a very difficult position to be in, and has a much greater impact, potential impact, than you've contaminated the roundworm samples. All right. I'll give you another example. We're interested in sequencing the Neanderthal genome. All right. Well, Neanderthals have been extinct for about 40,000 years. When we were developing the tools to sequence that genome of an extinct species, they didn't start with Neanderthals. They started with cave bear. And the reason they started with cave bear is it's relatively easy to distinguish cave bear gene sequences from human gene sequences. And if the sequences that were popped up from the sequencing project looked too human, you knew you had a contamination problem. It's very difficult to distinguish Neanderthal and modern human. And so if you had contamination, literally, you wouldn't be able to tell. All right. So we perfected the techniques, more or less. We tried to perfect the techniques by working in a case where it was easy to tell contamination and non-contamination. In the samples we're talking about in this week's news, if you've got fly samples that in fact are Doug, it's easy to pull that out. If it's my DNA that I'm trying to sequence and Colin's got his fingers in the tube, it's really difficult to see that contamination. And that's the implications of this study, is that when we're working with model species, there's scientific issues that could set us back. when we're working in biomedical cases, you could make the wrong diagnosis, you could make the wrong prescription, that kind of thing. And so what does it mean? It means we need to get better at the techniques that we use for screening the sequences that go in and try to eliminate these contaminations, because the bottom line is, if it's contamination from really closely related organisms, like one human to another, that contamination is really hard to see. All right. So, what do you tell your students about this? Well, you talk about modern genomics and biomedical genomics and the way that we're going to be using genes to diagnose disease, to diagnose drug resistance, to prescribe the correct drug for a correct disease. One of the complications that's going to come into this is this kind of contamination. And you just basically, you have to talk to your students about using these kind of techniques with the same open eyes that you use with any technique. There are always failings. Any technique that we have, there are problems with that. The better we understand the problems, the more able we are to avoid them. But this was a real sobering wake-up call, saying, hey, folks, we've got issues here with contamination. We can see it here. We know we're not going to be able to see it elsewhere. So what are we going to do now to fix things so that we move into the biomedical genomics? We're less likely to make these kind of errors.
We're 3 minutes away from the top of the hour, gentlemen. Colin, last comment goes to you today.
What would you like me to comment about?
Okay,
well,
I guess that's the show.
That was really cool. Like, the idea, I mean, we talked about before the idea that you're going to be sequencing your genes and potentially using that to make medical decisions. But it just struck me as Thomas was explaining it, the actual huge ramifications that has. If my doctor says, Colin, you've got Alzheimer's, but it's because the technician Kevin had his thumb in the tube to reverse your analogy here, I mean, that's a massive thing. And we're talking about how inexpensive gene sequencing will be. Well, if someone's going to tell me that I've got something like a very serious disease that's going to require treatment or whatever, I'm going to want that test done more than once, right? I'm going to want that repeated. And so it's going to have to be really cheap in order to get that done. We talked down, a couple $100 to sequence. But you know what I mean? If you have to get it done 4 or 5 times to ensure that you've actually got the right sequence, and again, there's just a lot of things that come out of there. So yeah, stuff I hadn't thought about before. That's very neat.
Not to be alarmist, I mean, any technique you use, any diagnostic technique you use, always has failings, right? And so
yes,
the important thing is not to find a perfect technique that has no failings, because you won't. The important thing is to understand the assumptions behind the technique you're working on and try to make the best decision you can. And so if that decision is you're going to take 2 independent samples, et cetera, that's fine. Just what we need to know is where are we making the assumptions and where are the potential pitfalls. So I think that the strength of that argument, the strength of that article is saying, hey folks, we've all known this is going to be an issue. Here are examples, concrete examples of these kind of contaminations. Let's keep this in mind and see what we can do to make this a little better.
Great article, Thomas. Thanks for bringing that forward. And I'm afraid we are out of time today. So on behalf of Colin Jagoe from the 4th Refinement Fuel Board and Dr. Thomas Merritt from Laurentian University, this is Kevin Cougler signing off with this week in Science and Education. Thanks again for joining us. We'll see everybody again next week. Have a good week, everyone.
See you, everybody.
See you guys.